Imagine sequencing the DNA of a patient. Let's say it's a young child with a really severe developmental disorder.
Right.
You run the whole exome, you scan every single protein coding region, you find literally nothing. The genes themselves are like structurally perfect.
Completely normal.
Yeah, no pathogenic mutations at all. But the child is still profoundly sick. And that exact clinical paradox happens all the time.
It really does. It's incredibly frustrating for clinicians.
Exactly.
Yeah.
And it happens because over 90% of the genetic variations that drive human traits and diseases, well, they don't actually live in our genes at all. They live in this sort of dark matter of the non-coding genome.
Yeah, it's the ultimate diagnostic blind spot. I mean, we've spent decades illuminating the coding regions, the actual genes. But when the mutation lies in the regulatory switches that are hidden in that non-coding dark matter, standard sequencing just leaves us entirely in the dark.
Which perfectly frames our mission for you today. Welcome to the Deep Dive. We are looking into a massive piece of research from the ENCODE Consortium, and this was published in Nature Methods. It's titled Multi-Center Integrated Analysis of Non-Coding CRISPRi Screens.
Quite a mouthful.
Definitely. But just to give you a sense of the scale here, this paper isn't just like one localized experiment. We are synthesizing data from 108 separate massive screens.
Which is huge.
Right. We are talking about over 540,000 individual CRISPR perturbations across roughly 25 megabases of the human genome. And the goal is to build the definitive rulebook for finding and controlling these hidden genetic switches, which are formally known as cis-regulatory elements, or CREs. Okay, let's unpack that.
Yeah, so to appreciate what the ENCODE Consortium has actually achieved here, we have to look at how we used to identify these regulatory elements. Historically, the genome has been annotated with hundreds of thousands of candidate cis-regulatory elements. They call them CCREs.
Emphasis on the word candidate, right?
Exactly. That word carried a lot of heavy lifting. It basically meant we were relying on associative biochemical markers to, guess where functional switches might be.
So is it just correlative data?
Just correlative, yeah. But this paper completely elevates the standard of evidence. They demanded functional causal proof, and they did that using CRISPR interference, or CRISPRi.
Okay, we should clarify the mechanism they're using here, because it's pretty elegant. They aren't using wild-type Cas9 to just, induce double-strand breaks and cut the DNA.
Right, no molecular scissors here.
Yeah, instead they are using a catalytically dead Cas9, dCas9. And it's fused to this thing called a K repressor domain. So instead of a scissor, it acts like a massive, intimidating bouncer.
I love that analogy, a bouncer.
It just parks itself at the target site. Right?
Yeah.
And it recruits heterochromatin-forming complexes and physically blocks the transcriptional machinery.
Just through sheer severe steric hindrance.
Exactly. It represses the local genomic neighborhood without permanently altering the underlying DNA sequence.
A highly effective bouncer. So the researchers deployed this dCas9-KRB complex across just a vast array of these candidate switches. And they heavily utilized the K562 cell line to establish their baseline functional rules.
Just to see which of these candidate switches actually repressed the target gene expression when the bouncer was parked on them.
Right, and when they isolated the truly functional CREs, this striking epigenetic signature emerged.
95.2% of those functionally validated switches perfectly overlapped with two very specific physical features on the chromatin.
Right, so first they sat within regions of highly accessible open chromatin. These are specifically called DNase hypersensitive sites, or DHS.
DHS, okay. And the second feature?
The second was a distinct histone modification called H3K27HC.
Okay, let's break that down for the listener. What exactly does that mean?
Sure, so that combination is critical. The DHS peak basically indicates that the chromatin is physically unwound, right?
Right.
It creates this accessible landing pad for transcription factors.
Like an open clearing in a forest.
Yes. Meanwhile, the H3K27 ACK mark, that's where the 27th lysine on histone H3 is acetylated. It biochemically neutralizes the positive charge of the histone tail.
Which means what? Physically?
It actively loosens its electrostatic grip on the negatively charged DNA backbone. So it facilitates a really active regulatory state.
Okay, but wait, I have to push back here for a second. You're basing this massive architectural rulebook largely on data derived from K562 cells.
Right.
But K562 is an immortalized leukemia cell line. It's known for having a highly rearranged, heavily mutated, and totally dysregulated karyotype. I mean, cancer cell chromatin is a mess.
It is a complete mess, yeah.
So how can we confidently extrapolate an epigenetic baseline from a cancer cell and apply it to a broad understanding of normal human biology? Like, did every single functional switch they found actually have both of these specific marks?
That is the exact right question to ask, and it's why the nuance in their findings is so critical here, because no, not every switch had both marks, and the exceptions actually validate the broader biological relevance.
Oh, really? How so?
Well, about 11.4% of the functional CREs had the H3K27X mark, but lacked the open chromatin DHS peak. And conversely, 8.6% had the open chromatin, but lacked the acetylation.
Okay, but what about finding a functional switch that was totally epigenetically silent? Like no open chromatin, no acetylation, just completely dark. Did they find any of those?
Exceptionally rare. I mean, out of the thousands tested, they found exactly 9 functional CREs in K562 cells that lacked both features.
Just 9.
Just 9. But here is the critical validation. When the researchers cross-referenced those 9 dark switches with epigenetic data from entirely different cell types,
Like which ones?
like WTC11 stem cells and GM12878 lymphoblastoid cells, they found that 7 of those 9 actually did display open chromatin in those other lineages.
Oh, wow. So the regulatory architecture is highly dynamic. And the K562 data isn't just some weird artifact of cancer.
Exactly.
So a regulatory switch might be like tightly packed away in your circulating T cells, but wildly accessible and acetylated in your liver cells.
Yes. What's fascinating here is that these specific epigenetic assays are not just correlative noise. They are profoundly predictive of actual real-world regulatory activity across different cellular contexts.
So just to do a quick recap for you listening, we've basically figured out the physical landmarks of these genetic switches, the DHS and the H3K27X marks.
Right, the roadmap.
But knowing that a functional switch sits in an accessible DHS region isn't really enough to manipulate it, right?
Yeah.
I mean, if we want to engineer therapies, we need precise physical coordinates. We need to know exactly where to park the DCAS9 bouncer.
And that requires optimizing the spatial geometry of the CRISPRi targeting. So the researchers mapped the precise efficacy of guide RNAs relative to the topography of the open chromatin, and they found that the repressor effect is not uniformly distributed across the enhancer region.
And concentrated, right?
Highly concentrated, at the exact summit of the DHS peak.
The paper actually models this out with incredible precision. If you visualize the accessible chromatin as a bell curve, the absolute maximum transcriptional repression occurs right at the peak of that curve.
Right at the top.
Yeah, and it's like playing darts. Hitting the edge of the board does a little bit of damage, but to really turn off the gene, you need to hit the absolute bullseye of the DHS summit. As you design guide RNAs that bind laterally, moving away from that center, the efficacy of the CRISPR bouncer drops off in a near-linear, highly predictable fashion.
Which dictates a fundamental rule for high-throughput screen design. You can't just indiscriminately target anywhere within a 500 base pair enhancer region and expect a reliable readout. You have to anchor your guides to the DHS summit.
But practically speaking, just throwing one dart, one guide RNA at the summit still leaves you vulnerable to biological variability or off-target effects.
Oh, absolutely.
Which introduces a massive statistical headache. If we're tracking these microscopic changes in gene expression, how many independent guides do we need to throw at an enhancer to confidently declare it functional?
Yeah, you don't want to generate massive false negative rates just by undersampling.
Exactly.
That was one of the most vital threshold questions the ENCODE team addressed. They utilized an immensely sensitive technique called CRISPRi flowfish, and they focused heavily on 2 well-characterized loci, GATA1 and HDAC6.
Okay, so they ran the numbers on those specific areas.
Right. By sorting millions of cells based on precise fluorescence-tagged transcriptional outputs, they were able to run a really rigorous statistical power analysis.
And they landed on a very specific mathematical floor, right?
Yeah, they did. They calculated that to achieve an 80% statistical power to detect a functional enhancer specifically, one that causes at least a 40% reduction in target gene expression,
Okay.
you must use a minimum of 13 randomly selected guide RNAs within that specific element.
Wait, 13 guides per enhancer. That is a heavy logistical lift when you are screening hundreds of thousands of candidate elements.
It is, yeah. But if you drop below that 13 guide floor, your statistical power crumbles. You risk missing critical phenotypic drivers just because your screen was mathematically underpowered.
So 13 is the magic number. But power is only half of the statistical equation, isn't it?
Yeah.
The other half is your baseline.
Exactly, the control group.
Right, because how do you distinguish the true repressive effect of your 13 targeted guides from the inherent chaotic background noise of a living cell.
You need a flawless control group.
This is where the paper challenged a major assumption in the field regarding negative controls. Because traditionally, when you run a massive pooled screen, you throw in a library of non-targeting guides.
Right. Basically, RNA sequences designed to be completely alien to human genome. They don't match any DNA sequence.
Right. So the dCas9 complex just sort of floats around in the cell, never physically binding to the chromatin.
But the alternative approach is using safe targeting guides. And these are meticulously designed to bind to regions of the genome identified as absolute genetic deserts.
Genetic deserts.
Yeah, vast intergenic expanses devoid of any known genes, transcripts, or regulatory elements.
Now, you would logically assume the biological effect of a guide floating aimlessly versus a guide parked in a dead genetic desert would be identical, right?
You'd think so.
And functionally, the paper found that the average effect on cellular fitness and transcription was the same between 1,000 non-targeting guides and 1,000 safe targeting guides. They both averaged out to a net zero effect.
However, the statistical variant, the spread of the data, was fundamentally different.
Yes. This was wild. The safe targeting guides exhibited a significantly wider variance. The standard deviation was 1.17 for the safe targeting guides, compared to a much tighter 0.86 for the non-targeting guides.
And if we connect this to the bigger picture, that mathematical variance is capturing a hidden biological reality.
What's actually happening to the cell?
Exactly. Even when you park a dCas9 KROG complex in a completely inert genetic desert, the sheer physical act of binding to the double helix, recruiting those heterochromatin factors, and altering the local 3D architecture, it induces a baseline level of cellular stress and structural noise.
Ah, so the non-targeting guides completely failed to capture that mechanical stress because they never actually touched the DNA.
Precisely. So if you use them as your baseline, your mathematical threshold for what constitutes significant repression is artificially low. By adopting safe targeting guides as the rigorous standard, you elevate the signal-to-noise ratio required to call a hit.
Which dramatically reduces false positive discoveries.
Which is absolutely essential when you consider the data ocean these screens generate.
Let's do another quick recap before we move on. So we have the epigenetic map. We know to target the DHS summit. We have our 13-guide mathematical floor. And we've calibrated our baseline variance with safe targeting controls. But once you execute all of that across 25 megabases of DNA, the analytical challenge shifts to the software. You need computational models that can reliably sift out the true regulatory hubs from the noise.
Yeah, the bioinformatic bottleneck. So the researchers set up a rigorous benchmarking showdown between 5 widely used analytical frameworks. There was AgriDissect2, CASA, CRISPR-Surf, MAGEC, and Relicus.
A real heavyweight bout. And
the performance delta between these algorithms was shocking. Specifically, when they stress-tested them against imperfect data, the researchers purposely spiked the dataset with low-specificity guide RNAs.
So guides known to exhibit promiscuous off-target binding elsewhere in the genome, generating artificial transcriptional ripples. They basically wanted to see which algorithm would take the bait.
Precisely. And an algorithm like AgriDissect2 completely hallucinated under those conditions. Its false positive rate just exploded. It jumped from calling 21 functional CREs to suddenly identifying 68 CREs.
Wow, so it was wildly overinterpreting the off-target noise as true regulatory activity.
Yeah. But CASA barely flinched. The number of CREs identified by CASA remained remarkably stable, despite the massive injection of off-target noise.
And the reason CASA won this benchmark comes down to spatial concordance, right?
Exactly. CASA doesn't just look for a drop in gene expression. It requires that multiple independent guide RNAs targeting the exact same tight, localized window of chromatin all independently verify the repressive effect.
It demands biological consensus. It's a highly conservative, probabilistic model.
And in high-throughput genomics, conservative modeling is really the only way to operate. I mean, if a pharmaceutical company is going to invest hundreds of millions of dollars developing a small molecule or an epigenetic editor to target a non-coding switch,
they need absolute certainty that the switch is real, not just a mathematical artifact hallucinated by an oversensitive algorithm.
Right.
But here's where it gets really interesting. While they were utilizing these advanced models to refine their targeting parameters, the researchers stumbled onto a subtle, almost weird mechanical quirk.
Oh, the strand bias.
Yes. It's regarding how CRISPRi interacts with the fundamental structure of transcribed DNA. Can you explain this?
Yeah, this is perhaps one of the most counterintuitive findings in the entire study. They specifically looked at guides designed to target regions within the actual transcribed body of a gene, intergenic CREs.
Now, logically, since we're using a bulky Dcath9K grab complex to induce steric hindrance, you'd think it wouldn't matter which strand of the double helix we target.
Right, it's huge.
The complex is physically massive enough to block the machinery regardless of whether it's parked on the coding strand or the template strand. So why does the paper highlight a highly specific strand bias?
Because the local physics of transcription matter just as much as the biochemistry. The paper revealed that CRISPRi efficacy is significantly higher when the guide RNAs are anchored to the non-template strand, which is also known as the coding strand.
Okay, let's visualize the mechanical environment here for everyone listening.
Yeah.
When a gene is actively being transcribed, the molecular bulldozer doing the work is RNA polymerase.
That's a great way to picture it.
It forcefully chugs down the template strand, unzipping the helix as it goes.
Right.
If you engineer your guide RNA to bind the Dcas9 bouncer directly onto that template strand, you are putting it dead center in the path of that bulldozer.
And RNA polymerase generates immense mechanical force. It possesses the kinetic energy to physically dislodge the Dcas9 complex from the template strand.
Just shoving it out of the way.
Exactly, stripping it away before the KRIB domain has time to recruit sufficient heterochromatin-packing proteins.
But if you design your guide to target the complementary coding strand, the RNA polymerase bulldozer largely bypasses the complex as it reads the template. The Dcas9 bouncer remains anchored, avoiding mechanical eviction, and successfully induces sustained local repression.
And this specific strand bias is a crucial operational parameter for future screen designs. Because if an investigator is scanning an intergenic region and unknowingly utilizes a library heavily skewed toward template strand targeting,
They will observe drastically reduced repressor activity.
right. And they might falsely conclude that the target region lacks regulatory function, when in reality, their repressor complexes were simply being mechanically bulldozed off the chromatin. You have to account for the transcription vector to ensure you aren't mistaking mechanical failure for a biological negative.
Wow. Okay, so let's step back and look at the whole architecture of the toolkit this paper has handed us. We began with this vast, undifferentiated expanse of non-coding dark matter. The ENCODE team has now given us the epigenetic coordinates, the DHS summits, combined with H3K27X marks.
They've established the statistical floor of 13 guide RNAs per element.
They've defined the mathematical necessity of safe targeting baseline controls.
They've proven that conservative spatial concordance algorithms like CASA are mandatory for filtering the noise.
And they've even mapped the mechanical strand bias for intragenic targeting. So what does this all mean for the future of research?
It means the barrier to entry for exploring the non-coding genome has fundamentally collapsed. I mean, the researchers didn't just publish the theoretical rules. They applied them to build a staggering open-source resource for the global scientific community.
Wait, they actually built the libraries?
Yes. Using these optimized parameters, they pre-designed comprehensive CRISPRi libraries targeting 3.2 million candidate CREs across both the human and mouse genomes.
3.2 million precisely calculated coordinates. That is incredible. They integrated the DHS summit topography, filtered out the off-target liabilities, controlled for the strand bias, and just published the exact sequences required to functionally interrogate nearly every suspected regulatory switch in our biology.
And this raises an important question for you listening right now as we consider the horizon of genetic medicine. We open this deep dive with a clinical paradox, right? The perfectly sequenced exome in a profoundly sick patient. For decades, we have been largely restricted to reading the genome like a static text. We're capable of identifying typos in the structural genes, but incapable of comprehending the hidden regulatory syntax that actually controls them.
It's like we knew all the words, but we couldn't see the punctuation or the formatting.
Precisely. But with this highly optimized, systematically validated toolkit, that paradigm is over. We now possess the functional capacity to systematically map, test, and eventually manipulate the 3.2 million non-coding switches that govern cellular behavior.
So we are moving beyond merely observing genetic architecture. We are acquiring the exact coordinates required to intervene in the regulatory dark matter.
Exactly. So here's something to think about. What happens to our approach to complex disease, to oncology, to developmental biology, when we finally transition from just reading the DNA to actively debugging and rewriting the operating system of human biology?
The dark matter isn't an impenetrable void anymore. We have the map, we have the coordinates, and the flashlight is officially turned on. Thank you for joining us today, and we'll see you next time on The Deep Dive.